Hello and welcome to Science Research Weekly, Season 5, Episode 13, Attack of the 50-Foot Octopus. Get ready for giant predatory Cretaceous octopuses, ice water clouds on a distant Jupiter, antiviral acrylic surfaces, 3D printing inside living cells, March's top 40 new CRAN packages, and a grant on engineering, biological, and biomedical systems. This material will cover from April 24th to April 30th. I was out last week for a space-related meeting, so we'll just jump right in and get started with some research articles. First up is compositional selection of phospholipid compartments in icy environments drives the enrichment of encapsulated genetic information. So this paper talks about the lipid world hypothesis that actually lipids, or the intercellular components and the chemical composition of the membrane, can actually act as heritable information. And one thing that they explore here is that maybe freezing and thawing cycles actually help with that. So very interesting set of concepts coming together. Next up, earliest octopuses were giant top predators in Cretaceous oceans. So the summary, that editor summary, is more or less that the Kraken is sort of real, at least in the Cretaceous. Researchers found, I believe, a jaw of an octopus that might have been up to 19 meters in length. And the real amazing picture here is figure 4, looking at body size estimation of late Cretaceous octopus. You can see them, the finned octopus being larger than a mosasaur. So if that's true, that would mean giant octopuses as top predators. All right, next up, a second visit to Ebs Ind Ab with JWST. New photometry confirms ammonia and suggests thick clouds in exoplanet atmosphere of the closest super-Jupiter. So super-Jupiters, or cold Jupiters, are a little rare. Hot Jupiters are more easy to spot. But they were looking at this cold giant planet, and they actually believe that there is more water ice clouds than we typically think for cold Jupiters. And so that complexifies how these planets are actually formed, and the diversity of chemical elements which actually can make up their atmosphere. All right, moving along, we have designing scalable Mechano-Virucidal nanostructured acrylic surfaces for enhanced viral inactivation. So antimicrobial surfaces are kind of a big thing, but you can also develop antiviral coatings. And so that's what they did here, using things like, looks like ultraviolet nano input lithography. And essentially they're able to create these very small surfaces. They're very rough, and they essentially pop them like balloons. That would be my best analogy. Let's see, do we have any images here that pop up? Okay, not at this time. But yeah, imagine a very rough surface that the virus can't actually get onto, this acrylic surface, that it will actually rupture if it tries to land on it. So very interesting use of surface sort of physics, surface chemistry, and medical applications. Okay, moving along. Removal of microplastics from drinking water by Moringa oleifera seed, comparative performance with alum in direct and inline filtration systems. So it turns out the plant Moringa, sort of used in, I think, traditional herbal medicine, turns out that using them, you can actually remove PVC microplastics. Now, it's not as efficient as other more highly intensive, more chemical, technological processes, but they seem to work for maybe more rural areas, more remote areas. In fact, I would say a use case for this would be something like space applications. Anyways, that's my going out on a limb here, but that is but on that paper. All right, final regular paper of this escapade, we have 2-photon 3D printing of functional microstructures inside living cells. So researchers are able to 3D print shapes inside living cells. And figure 1, if you're watching the video or if you want to look up this paper, kind of really shows it. They're able to insert into the membrane, have this photoresin injected, and then they can use this 2-photon polymerization method to actually create something in there. In fact, they have this little tiny elephant as one of the examples. Very cute elephant inside an actual living cell. All right, we will get to either the twilight zone or the danger zone, whatever analogy you want to get to, because we're going into preprints. We'll start with archive and then go into bioarchive. The one archive I found is called Can We Teach Generative Artificial Intelligence the Design Language of Engineered Living Materials? So looking at ontology and codification scheme for engineered living materials. And I hadn't really thought about engineered living materials, but they break it down. For example, table 1 is very interesting, has the different taxonomic ranks and the different descriptions. We have archaeal, ELMs or engineered living materials, even on plant or cross-kingdom ones. So yeah, just the concept of engineered living materials and sort of classifying them is novel and something I didn't encounter before. But then adding on, well, not only do we have this here, but can we teach this to AI so we can use AI models to in this subtopic is yet another layer. So I think this is a preprint worth going into more. Okay, switching over to the 2 bioarchive. The first one is harnessing AI to build virtual cells. So we get AI again. Looking at modeling virtual cells. Virtual cell modeling is really helpful for predicting cellular processes, simulating cellular processes. So the system they're talking about is VCH or VC harness, autonomous AI system to construct perturbation response models. And then finally, we have urban soil microbiomes exhibit taxonomic and functional potential for enhanced contaminant cycling. So urban microbiomes in the soil are going to be different from other places. And so they were able to use this global urban soil environment ecology network and examine things. And they did find greater spatial variability in microbial taxonomy compared to functional potential for these urban microbiomes, as well as other findings. All right. Let's switch over to some research tools. Over in the R world, R 4.6.0, codename Because It Was There, has been released. Then on our bloggers, we got a couple today. One of my favorites, the 20 top 40 new CRAN packages for March 2026 is up by Joseph Rickert. So you can look through there of what he considers the top packages of over 300. Then there is Little Useful R Functions, a desk plant simulator by Tomaz SQL. So a little simulation game with twerks and hidden gems and ASCII art, growing a little R plant. Then finally, we have from Seascape Models, Design Your Plots. That is ggplot for mobile. So some tips about keeping mobile in mind with plot development, plot generation. All right, new R packages that I found that might be useful from this last week is R pandas, translating from R to Python's pandas packages, or package. All right, closing our time here, we got some grant opportunities. The first one is from the National Science Foundation, Engineering, Biological, and Biomedical Systems. And that program is to expand understanding of biological and physiological systems to improve human health. So that might be of interest for listeners. There's also from the Advanced Research Projects Agency Energy, inspiring generations of new innovators to impact technologies in energy, also known as Ignite 2026 with two eyes in the middle. So it is, in development of transformative science and technology solutions to address energy and environmental missions. And finally, from Grant Forward, there is a John Templeton Foundation grant now out for the life sciences. Especially trying to answer questions like what constitutes life, whatever its limits, what are core processes, and etc. So if you find that interesting, that is available as well. With that, that ends our Science Research Weekly for this week. Thanks for joining me as always. And as always, science on.